In recent years, the field of genetic engineering has witnessed incredible advances, none more so than the groundbreaking CRISPR technology. Standing for clustered regularly interspaced short palindromic repeats, CRISPR has revolutionized our ability to edit genes with unprecedented precision and has opened a new frontier in the realm of genetic modification. But as with any powerful tool, it carries with it a host of ethical and technical questions. Today, we delve into the dual specter of promise and peril surrounding CRISPR germline edits. The capability to edit the germline, essentially manipulating the genes in sperm, eggs, or embryos to affect future generations, heralds potential that was once confined to the pages of science fiction. Among the prominent promises is the ability to prevent hereditary diseases before a child is even born. Conditions such as cystic fibrosis, sickle cell anemia, and Huntington's disease could potentially be eradicated, offering hope to countless families afflicted by these inherited conditions. Yet, this scientific leap is not without its controversies. Enter the case of He Jiankui, a Chinese scientist whose experiment in 2018 sent shockwaves through the global community. He Jiankui announced the birth of twin girls whose embryos he had genetically edited using CRISPR to confer resistance to HIV. His actions resulted in widespread condemnation, primarily because the scientific community deemed the experiment premature and reckless. It was conducted without adequate understanding of the long-term effects and limited transparency, and raised serious ethical concerns about consent, the potential for unforeseen health impacts, and the broader implications for humanity. One of the significant technical challenges with CRISPR is the potential for off-target effects. These occur when CRISPR makes unintended changes to the genome, which could lead to harmful mutations, cancerous growths, or unanticipated side effects passed down to future generations. Such risks underline the necessity for rigorous caution and remind us why many scientists advocate for a moratorium on germline editing until these issues are comprehensively addressed. Globally, the response to germline editing has been mixed, with regulatory stances ranging from cautious prohibition to a call for more stringent controls. The United States, for example, currently prohibits the use of federal funds for research on viable embryos, reflecting a more conservative approach. Meanwhile, countries like China and the United Kingdom have adopted more permissive but closely regulated frameworks, demonstrating a willingness to cautiously explore this controversial terrain. This inconsistency underscores the need for international collaboration and dialogue to establish ethical guidelines and ensure that advancements are guided by both scientific integrity and societal consent. As we navigate this complex landscape, it becomes imperative to consider the ethical dimensions. Should we, as a society, prioritize disease prevention over potential risks? Or does the specter of unchecked genetic modification require us to proceed with greater restraint? The answers are not straightforward and require a nuanced understanding that balances scientific potential with moral responsibility. In conclusion, CRISPR germline editing stands on the precipice of remarkable possibilities, promising significant advances in disease prevention and human health. However, it also poses serious ethical and technical challenges that demand our careful consideration. As we advance, it is crucial that public debates remain respectful and informed, exploring these profound questions together. Your thoughts on this matter are invaluable, so we encourage you to respectfully share your perspectives in the comments below. Let's ensure that as we edge closer to this tomorrow, we do so with wisdom, compassion, and a shared commitment to the betterment of all humanity.